Hello everyone, my name is Jeongbin Park. I'm a PhD candidate in bioinformatics. Today I'm going to talk about a presentation about single-cell foundation models as physical AI. As you can see in the left plot, the current size of the pharmaceutical market is about $1.6 trillion, and it is expected to grow. Very recently, U.S. Food and Drug Administration releases ISTAN, which implies the decrease in animal experiment, rather increasing organ-on-a-chip, machine learning model when AI to replace with animal experiment. So we must build predictable data-driven biology instead of animal models. And the data is ready. In 2021, some corpus data such as tabular sapiens has about 0.5 to 1 million cells, and now we are dealing with 1 billion cells, such as Chan Zuckerberg Initiative Billion Cells project. Some computational biologists pointed out as a human is composed of 60 trillion cells, we must also approach that trillion cells to address the biological heterogeneity and complexity. But the problem here is the unstructurization of the data. The format is very different. The annotation can also be diverse. So to address that unstructurization of the data, so many scientists pointed out the large language models. Jeffrey Hinton, the Nobel Prize laureate in physics in 2024, said this new form of AI excels in modeling human intuition rather than human reasoning. So the LLM, which is to predict the next words given the sentences, can then be a good tool to address the unstructurization of the data set, as I mentioned in the earlier slide, in the form of human intuition manner. Just like the large language model, the hot topic these days is the physical AI, which is to predict the next physical events given the physical phenomena. And also the one I'm mentioning today, the single cell foundation model where virtual cells can also be an example of the physical AI because it predicts the next gene expression given gene expression corpus data. And now I'm going to talk about my paper, Cell Llama, which is about a foundation model for single cell and spatial transcriptomics by cell embedding, leveraging language modeling abilities, which is published in Advanced Science in 2025. This is a novel architecture that treats single cell expression profiles not as mere numerical matrices, but rich contextual sentences. Let me explain further more about my algorithm. So for example, each cell is composed of multi-dimensional gene expression matrices, and the dimensionality corresponds to the number of genes. Instead of dealing with the quantitative values, we can translate the cell into a sentence as can be seen here, based on gene length embedding. And that sentence can be transformed into 384-dimensional vector by using a sentence transformer. And that multi-dimensional vector corresponding to each cell can be applied in diverse bioinformatics downstream analysis. Let me give you some examples. So there are a lot of batch effect in biological bioinformatics data, and that batch effect should be addressed. For example, when you perform the differential gene expression analysis, a certain gene is preferentially expressed in a certain data set, but that discovery is not biologically meaningful. So that batch effect can be eliminated in diverse manner. Some algorithms are quantitative algorithm, and the other ones is basically addressing some specific approaches just like mine. So for example, as I mentioned, my algorithm Llama translates itself into multi-dimensional vector without consideration of any quantitative measures. In such a case, the batch effect, which is inherent in that quantitative values, can be naturally eliminated so that we can easily incorporate two different data sets into a very harmonized manner. And also, if we know more information about a certain data set, which is called metadata, then we can also integrate that data set into more biologically meaningful way. And please note that metadata is basically natural language description, while gene expression data is somewhat different modality, but that data can be incorporated into a single embedding space. That is very important point of view in this algorithm. Also, This is another application. The reference data set has a kind of cell type label, and the test set, we want to know the cell type labels. That is a situation I'm thinking about this in this application. And as I mentioned, each cell can be co-embedded no matter what, whether they are come from which data set. So each cell in the test data set is now looking for the nearest reference data set to identify the corresponding cell type, as can be seen here. And we can easily and automatically transfer the labels in the reference data set into the test data set in a way of nearest neighbor embedding. And please note that as we consider nearest neighbor in the embedding space, as I mentioned, 384 dimensional embedding space, we can also estimate the distances. So for example, the higher distance, the more uncertain. That uncertainty can also be measured in this way, and uncertainty can be shown in diverse forms like transitioning cell types, developing cell types, or technical artifacts, and so on. This is another application, even not only for single cell data only, but also the spatial transcriptomics can be also analyzed by the help of Zellama capability. So for example, the spatial information and the context can also be incorporated into the form of metadata and also natural language description. And these natural languages can be transferred into 384 dimensional vector. As for example, here, fibroblasts can also be subclassified into different clusters based on the spatial context and the niches, which correspond to the cell type composition around it. As we can translate the biology into language, we can also generate it. The next phase utilizes a language-based formation foundation models to simulate how cells will react to specific drug interactions and genetic knockouts, entity in silico, and also physical AI or lab automation as well. Thank you, and I'm welcome to any question.